Hey everybody, this is Katie Zander from the Standards Coordinating Body. Today we're going to have Simona Sarkar from NIST talk to you about the new tool, which is the Introduction to the Cell Counting Method Evaluation Tool, also known as COMET. And so I will let Simona take it away.
Thank you, Katie. I'm Simona Sarkar again from NIST, the National Institute for Standards and Technology in the Biosystems and Biomaterials Division. I have served as an expert in ISO TC 276, which is an ISO committee for biotechnology. And in that committee, I was the project leader for the ISO standard 20391-2, which is a cell counting standard, and that was published in 2019. And I've also worked with my team here at NIST to co-develop this COMET tool to help in implementing the recommendations that are outlined in this standard. So COMET stands for Counting Method Evaluation Tool. And as I said, it was developed to analyze the performance of cell counting methods based on a dilution series experimental design. And this tool calculates the quality indicators for cell counting methods that are described in this standard. So this standard outlines a dilution series experimental design and statistical analysis approach to actually quantify the performance of a counting method and eventually compare the performance of counting methods. And it's mostly applicable when you don't have access to a reference material that can establish true accuracy of a method. And these quality indicators are based on the principles of repeatability of a measurement, as well as proportionality to the response to dilution. And you can learn more about that standard at the ISO website, as well as our NIST website. And we'll give you some resources for that. So this ISO approach that's available is very useful in counting method development, optimization, and validation and qualification. The tool itself, the COMET tool, can be used to perform the statistical analysis that's described in the ISO standard, and it also provides the information needed to meet the reporting requirements that are outlined in the standard. Additionally, it compiles metadata associated with the experiments conducted for this evaluation, and it provides some additional analysis of count and viability data as well that users might find helpful. So right now, we can take you through that tool and do a quick demo to give you an introduction to the tool itself. So here we have the counting method evaluation tool, and it's actually written in Shiny, which is an R-based platform. And the tool itself, as you can see here, has a few input fields. I'm scrolling through them right now. In the beginning of this tool, we have provided several features that can be downloaded that can help introduce you to the tool. So all of the instructions, templates, example files can all be found in this original drop-down menu. So for example, in the README file, this is actually an Excel file that provides details on how the tool can be used and all of the templates and a description of all of the fields that are in the templates. And also it provides additional resources to help you understand how to use the tool and some papers and publications that can also support your understanding of the standard itself. So again, as I mentioned, this tool is based on a dilution series experimental design where you start with a stock cell sample, you dilute those cells into different concentrations, and we use that dilution fraction that is well controlled in order to ask about the quality of the measurement. So following this dilution study, which is outlined in the ISO standard, we can derive quality indicators through statistical analysis, and that's where COMET really comes in. It automatically takes in the data provided and outputs the quality indicators that are described in the standard. And also it collects and outputs the metadata that is used for reporting. So you can see here, this is an example of the template that's used. It's got different columns with different headers for different aspects of the data. So the raw counts, which replicate observation you're looking at, things like that. And we do have a description of what each column means and what kind of information needs to be entered in each column. In the case that you don't want to analyze all of your metadata or don't have it available, or simply don't want to, switch over to that full template, we do offer a more simple template that can still accept data that's a little bit more intuitive to how we might be collecting our cell counting data. So for example, for a method, you can have your different target dilution fractions and your observations of cell count at your different observations of cell count across your different replicate samples. So there are different options to be able to enter data depending on what you might have available to you. And then also this tool, this README file goes on to describe the user interface and all the different input parameters that you need to provide. And it also describes each of the output tabs. Finally, you have also example data sets embedded into this README file, but you can also get those example data sets directly from the COMET tool. So right now we're back at the COMET tool, and you'll see that you have example templates and also empty templates that you can download and enter your information into. So right now we can go ahead and go through a quick example of running this application, running this tool. So I've already downloaded one of the templates from this drop-down menu. So I'm going to load it here. And so once I load that template in, I can select these different features. And as you'll see as we go through, everything pretty much has a default value. So you can go through and not change almost anything and still get an output that is following the recommendations of the ISO standard. So for example, here, denoting my missing values, in my template, I should be using the letters NA, or if I use something else, I can indicate that as well. So I'm going to stick to the defaults for this demo, but you can also change you have to establish a mean variance relationship according to the ISO standard, but there is a default that is kind of recommended in the standard and also the default in this tool, but there are other options that you can follow through with. Same thing with smoothing. This is one aspect of the standard. There's a default, however, you can enter your own. We also have Bootstrap Analysis to help provide confidence intervals for our metrics that we generate. And you have an option to select that. For kind of test runs, we recommend about 50. For a final run, you might be closer to 1,000 runs, or you can enter a custom number. And you can also set the confidence interval that you'd like to use. We finally and importantly is the actual proportionality indices, which are the main output of this standard that help you to understand the quality of your cell counting method. And we have different options for doing this as outlined in the standard. And there is a recommended one, mostly because it has some sense for cell counting, but I also personally like to look at this smooth scaled sum of absolute error. So I'm going to select that as well. And now I simply can hit run analysis. So as you saw, really I had to change very little in order to get this analysis started. So now, looking at the data, it's been uploaded. You saw that toolbar that said it was going through our Bootstrap analysis. Here's the data overview tab. The first figure here is the raw data. So in this tab, you can see all of the different observations and the replicate observations and samples going through all of these. So in this case, we're comparing 3 different cell counting methods, method 1, 2, and 3. And we can see that the different methods actually produce what looks like a different cell concentration. And you can also see some increased variability. And we'll learn through this analysis if there is some loss of proportionality in these methods as well. So this is the raw data. And I will note that this is full template example number 3, which is data from a publication that is available. And so from this plot, and as you'll see here, we also have a pretty good description of what each plot is trying to describe. So next we've got a plot here that actually fits the proportional model to the data. And now we've averaged across replicate observations. So you can see mean cell concentration here. And so Now we're starting to get a better picture of our data. And then this also depicts the residuals, and we use these residuals to calculate our proportionality index. And you can see raw residuals, smooth residuals, as well as scaled residuals. And looking at, for example, the scaled residuals, which is what we're going to be using in our calculation, you can already start to see some differences in these methods, with method 3 seeming to have less of a tight interaction with the proportional model fit, which is represented by the horizontal line at 0. So this is our data overview. Then, importantly, we've got our actual quality indicator. So the standard requires that you report the R-squared value, and so these are depicted here for the 3 different methods. The confidence interval here, it'll describe how that confidence interval was derived. In this case, this is based on the Bootstrap analysis. We also have the proportionality indices here. So this is where we look at, is there a real difference in the way these methods respond to a proportional dilution fraction series? And we can see that yes, in fact, method 3 does seem to have a significant deviation from proportionality in both of our metrics of proportionality. And the standard also requires that you report the mean concentration for each of your samples, and this helps to understand the comparability of proportionality indices from one study to the other, and also the coefficient of variation, and this is giving a sense of precision between the 3 different methods. Finally, we also have here the proportionality constant, which allows someone to understand how your proportional model fits were modeled, but it also provides information on bias between methods. So the proportionality constant is actually equivalent to the prediction of your stock cell concentration based on each method. So you'll see that method 1 seems to have a higher estimation of that stock cell solution than method 2 or method 3. And so there does seem to be a bias between these methods. Moving forward, these images are great, but in order to be able to report these metrics, you need the actual values as well as these kind of errors and confidence intervals. So all of that information is here in table form. So you'll see that for methods 1, 2, and 3, you've got your mean concentration and all of the intervals around that. You've got the CV, the R-squared, and the proportionality indices. And a very useful feature here is that you can download all the tables, so you will get all of your information in Excel format so that you can create a report as needed. And also, take the data into other analysis programs. Another important reporting feature that's required by the standard is to actually describe the experimental design that was conducted when in creating these indices. And so here you can actually look at, based on the data that was entered, the structure of the data that was entered, this tool recreates what your experimental design was. And so it's good for 2 things. It's good for troubleshooting, whether or not you've entered your data correctly and it matches your expectations, but also it allows you to, again, export this information so you can easily generate a report that describes your exact dilution series study. So you have pipetting error, how many replicate samples, how many replicate observations for each of your different dilution fractions. So if you had an error and you missed one of these, or you had a sample handling issue, these numbers would change, and it might say that you only had 2 replicate observations instead of 3, and that's all captured here. Also required for reporting is what kind of statistical analysis did you do? And so here we have the actual equations that were used for the modeling of the proportional model fit, as well as what your proportionality constants were in this table. Based on my selection of which proportionality index type I wanted to calculate, it gave me the equations for both of those, and it described our smoothing approach as well as our number of iterations. So this is, again, an important feature to be able to capture all of the details of how this was done. Now, this is something that isn't directly described in the standard, but it's a very useful tool for our users. It's comparing methods. And so here you can see that we can directly look at the different comparisons of our proportionality indices. And you can see that method 1 compared to method 2, here's the ratio to achieve that comparison. And we actually explain based on what this ratio value is and the upper and lower bootstrap analysis, if we see a significant difference or not. And so you'll see here that in some cases, yes, there was a significant difference in the proportionality index between methods. We've also got some other interesting visualization tools to help interpret the results of this type of dilution series study with descriptions of how to interpret these plots. So there's a few of these other kind of interpretation plots. And also there's the bias comparison table. So this uses that proportionality constant now to ask if there is a bias between our counting methods, and it tells you if this is significant or not. Also, as a part of this tool, if you are able to enter the kind of metadata associated with your cell counts, for example, the time of execution, so here's time elapsed. If you enter that into your data table, this will allow you to visualize how your sample integrity held up during your experimental design and troubleshoot any problems that might be occurring during that study. So this gives you some different display methods and things like that. Also, if you do have some measured dilution fractions that you conducted, you can look at your pipetting error. And interestingly, you can also look at viability over time. And this allows you to monitor, again, the integrity of your sample. And in some cases also gives you a little bit of a window into comparing those viability results. And finally, we do have a little bit of push in the viability direction. So if you have viability data that's been entered into your template, you can visualize that viability data. So for example, here you've got our 3 methods, and method 1 is reporting higher viabilities than the other 2 methods. But also interesting in this visualization is the distribution. Technically, all viability should be the same, even if the concentration is different. But you'll see there's some spread in that viability value depending on what concentration you were in. And so we actually provide some statistical analysis of this and ask ourselves, is there an actual difference in these different viability measures from the different devices? So that in a nutshell is what this tool is and what it offers. There's some other download results buttons here that allow you to download everything in just one Excel file, but it's not going to be ordered perhaps as conveniently as some of the other download tabs. So, you know, there are several developers of this tool. We have technical contacts as myself and my colleague Laura Pierce. The contacts for the statisticians who created this tool and also helped to develop the statistics used in the ISO standard are David Newton and Steve Lund. And so we hope that this tool is going to be useful for people that are trying to implement the ISO cell counting standard. And you know, we recognize also that there are other tools that instrument manufacturers and vendors might create that have other capabilities that this tool doesn't. For example, you know, we don't have as great version control or kind of all the software in place to do a lot of tracking for GMP and things like that. But this is an open access tool that can be used to appropriately conduct the statistics that are described in the ISO cell counting Part 2 standard. So with that, I will stop sharing my screen and allow Katie to take
I'm going to share my screen again. And if I could ask you to just point out the resources again, because I wasn't on that slide when you did it. We'll have that, and then just in closing. Does that work?
Sure.
Thank you. And that was great. Okay.
Thank you. So we have several resources available in order to help in understanding and implementing this ISO standard. So the first resource is obviously this COMET tool. which is listed here. Also the ISO standard, which is available. And we also have a NIST resource page that describes some of the theory behind the ISO standard and provides some more resources along this. So we are excited for people to use this tool, and we are happy for folks to contact us to learn more about it, or if they need any help in the implementation. And we are still, happy to have feedback on the use of the tool itself and any features that can eventually be added to this tool or changed around on it to make it more useful for everybody. Thank you very much.
Thank you very much, Simona. And if you would like any more information that isn't on any of those resource pages, please feel free to either email Simona directly or to email me over at SCB, and I can help put you in touch with who you need. And also keep your eyes open for the standards implementation courses that we're going to roll out later in 2022 about these ISO cell counting standards. So thank you very much, and thank you so much, Simona.
Thank you as well.